Okay, Lecture 7. Lecture 7's topic is PCR. Of course, PCR stands for polymerase chain reaction. So what is PCR? You probably learned about this topic a while ago when you took your Biology 100 class. Probably there was a discussion about this topic. So PCR, polymerase chain reaction, means what? It's basically DNA replication that's happening inside of the test tube. So normally DNA replication happens within the living cells, especially within the nucleus where your chromosomal DNA is located. But if you can create the same environment inside of the test tube, of course you can replicate that process. So you can make DNA copies within your test tube. That's what it is. And why is it important? Because it is used in many different places. You have a tremendous amount of application. Okay, so it can be used for detection of DNA. Small trace amount of DNA can be amplified, so you will have more DNA made, so you can use that for different types of analysis. Think about forensics. Okay, maybe small evidence what you can find from the crime scene. It could be a drop of blood, or could be a drop of bodily fluid of any kind. From there, whatever the small amount of DNA you can find, you can replicate, so you can make a lot more copies, so you can do some analysis on that DNA, and that analysis results can be used in solving crime scene investigation, right? So that's one good example. Okay, and also you can use that the process of DNA cloning, gene cloning process. Also, it is used in the DNA replication process. Also, it is used in DNA sequencing process. Also, it is used in screening the gene expression pattern using a process called RT-PCR. In that case, RT stands for reverse transcription PCR. We'll talk about that, what it is. And they're used in detection of DNA, and especially viral DNA. You know, these days, you know, the COVID-19, when they do the screening, they use PCR process. You know, when they do the swab of your, you know, nose, you know, the throat, whatever the mucus they get, and then they use a specific set of PCR primers that can amplify out the presence of viral DNA in the sample. So detection of viral infection can be done using PCR technique. And prenatal diagnosis to screen for any kind of you know, malformation in your fetus. Things like that could be done. And detection of tumor, early stage tumor, or cancerous genes, they can be done. Okay, especially in the field of anthropology or archaeology, when they use a small sample of DNA isolated from ancient ruins or dead bodies or whatever. You can make more copies of that and use it for analysis. PCR is also used for process of mutation, especially used in site-directed mutagenesis. What does that mean, site-directed mutagenesis? That means you can change a single codon that codes for a single amino acid within a protein. So you can change a single amino acid within a protein using this process and see how that changed the structure and function of a protein, right? So the bottom line is PCR is used in many different places. That's why the inventor of this process, Karen Mullis, won a Nobel Prize. So how does PCR work? Okay, so in order for you to understand that question, the answer for the question, you need to understand how does the DNA replication process work inside of the cell, right? If you remember the DNA replication inside of a cell, what do you need for you to be able to carry that out? So of course you need the original DNA molecule, so which is made out of 2 strands. So anti-parallel 2 strand, 5' to 3', 5' to 3', and those 2 DNA strands should be come up open. Okay, so there should be a protein or enzyme that makes DNA template strand open. So the DNA helicase would do that, and then there's an enzyme that can make something called a primer. Okay, RNA primer will be made. So that's a primase enzyme. So the primer should be synthesized. So there you go, RNA primer, that's made right there, and that is done by an enzyme called RNA primase. Once your primers are made, then DNA polymerase will grab the tail of the primer and start its replication process. So DNA polymerase is used, and the nucleotide will be needed to start the synthesis of new strand. Okay, so a lot of enzymes are needed. The nucleotides are needed and all those things. So it's kind of complicated. Okay, so then how would you recreate that environment? How would you collect and provide all these enzymes and things instead of a test tube? It's a kind of challenging thing, right? So you can provide the minimum condition actually. So let's see how it works. If you copy and paste this website address in your search engine browser, for example Google or something, okay, here's the website. I just copy and paste it, and if you go there, yeah, you will see this website. Okay, it'll explain the process using this animation amplification process. Just click on that. Okay, so here's the minimum requirement. So the original strand of DNA, the double strand of DNA, the DNA molecule, right? So you need the enzymes to open that up, so helicase. But instead of adding that enzyme, you can use physical treatment such as high temperature. So at the high temperature, what's going to happen? So these two strands are held together through a lot of hydrogen bonds between the bases that are found from between those two strands, right? So, but the hydrogen bonds are pretty weak, so they'll be compromised, they'll be broken up if you expose this DNA strand to high temperature conditions. So strong thermal energy will break up all those high hydrogen bonds between two strands. So you can do that. You can expose this DNA molecule to a high temperature. So you're going to expose it to a denaturing temperature, which is about 94 or 95 degrees. About 1 minute will do that. So if that happens, see what happens. So yeah, double strand DNA becomes single stranded because all those hydrogen bonds are broken up, right? Once that happens, and then you're going to add the molecules, so-called primers. Primers are what? Unique single strand DNA that has a matching sequence to one of these two strand DNA strand template. Right, so you need two primers to bind on both strands. One primer may bind on that side, the other primer may bind on that side, and they have unique sequences. So they are two different primers, but they are both needed for PCR to work. So let's add primers. Yes, they added one primer from here to here, it bound. The other primer, it bound from here to here because they have the matching sequences. So we call that step as primer binding step, primer annealing step. Okay, normally that primer binding, primer annealing is done at a higher temperature. It's high enough so all these the hydrogen bonds are made, but it's not too hot, so the bonds would not be made. Okay, but that's a high temperature enough, so it's gonna only allow this specific binding, not non-specific binding will be compromised, broken up. So we'll talk about that later on, what that means. So once primer molecules are bound, then you can add the DNA polymerase, which will bind to the tail of the primer and continue the replication. So let's add DNA polymerase. See what happens. So DNA polymerase was added, and it grabbed the tail of the primer, and then it continued the DNA replication. One direction, another direction. So now at the end of one PCR cycle, what do you see now? You have two DNA molecules just made from a single template DNA molecule, right? But one important thing you can see is the replicated copies. Okay, so the replication only happened from the primer binding site and until the end of the end of the template DNA. So from here to here. Same goes on this primer bound there, and then replication started from there until the end. So these are not identical sizes. The template DNA was long, but the newly made DNA is not that long. Maybe only covers from the primer side to the end, primer side to the end. So okay, that's how it happens. Can you repeat that PCR again? Let's try that. So you have two DNA molecules. Now you're going to denature that by heating it up. Okay, So then you're going to add the primers so they will bind. So primers bound to all those DNA strands, right? And then you're going to polymerize by adding enzyme and DNA nucleotide. There you go. At the end of the second cycle, how many DNA molecules do you have? 1, 2, 3, 4. Okay. And how about the size of the DNA? So this was just the same as the original DNA that was just made after the first polymerization. Same goes with this one, right? But how about this DNA right here? So it's only replicate from here, the primer binding site, and until the end of its template DNA. So from here to here, that's it. And how about this one? Same way, primer bound there, and it went until this much. Okay. So now, you know, DNA replicate products, they have different sizes, right? How about if you repeat one more time? So denature it, heat it up, and then add the primers, and then the polymerase. And yeah, so that's the end of the third cycle. How many products do you have? 1, 2, 3, 4, 5, 6, 7, 8. 8 products, right? And look at the size of the products. From here to here, from here to here, from here to here, from here to there, here to there. Okay, seems like more and more DNA replicate products are kind of small between the two primer binding sites, right? Repeat that again. You get 16 products. Okay, after fourth cycle, you have those. How about after 5 cycles done? Look at it. How many products have the size between the two primers? Majority of them by now, right? So you continue that about 30 times. Okay. So if you do the 5 replication cycle, you have that many products made. But if you continue 13, 15, and continue all the way to about 30 times, 30 cycles, how many copies did you just make? So that's million, 10 million, 100 million. That's a billion, right? So 1 billion copies are made after 30 cycles from one single DNA template. So you can make so many copies at the end of it. Okay? Target copies. Okay? What does that mean? Those are the copies between the two primers. Okay? The majority of them are having the size between those two primers. Okay? Does it make sense? That's how PCL works. All right? So you can play that animation to help you understanding about the PCR process. Okay. So if you want to replicate your products, okay, what are the things that you need? Instead of using a bunch of enzymes, I told you what. Instead, you can use maybe a heat exposure, right? By heating up your test tube, okay, at like about 94 degrees, about 30 minutes, or 30 seconds, or 1 minute. That's enough, right? So the heating is the first treatment, okay? And then you cool it down so the primers will bind. So the second step is primer binding step. And at what temperature condition would that happen? So that depends on the sequence and the length of your primer, okay? So we'll talk about how you can figure out the sequence condition, okay, and the temperature condition. But the third step is the polymerization step done by the DNA polymerase. Okay, so it takes about 1 minute or 2 minutes at about that temperature, 72. Why is it 72 degrees? Because you're going to use a specialized polymerase enzyme that's isolated from extreme thermophile, some thermophilic bacteria. That enzyme is called the TAC, T-A-Q DNA polymerase. TAC, since it's isolated from an extreme thermophile that leaves the temperature about 72 degrees Celsius, that is actually the optimum condition for the TAG enzyme to work. Okay, so you use that temperature condition. So these are the 3 main steps where your in vitro DNA replication can happen. Okay.